Okay, thanks a lot for coming to the seminar. So we're going to start. So as usual, people on Zoom, we're waiting for your question in the Q&A tab rather than in the chat. And we will allow you to interact with the room during the discussion session. And with that, thanks Roger for introducing today's speaker.
Okay, thank you very much, Jeanne, and thank you very much, Christoph, for coming to Montpellier. So it was with pleasure that we invite Christoph Grunau, who is, by the way, also head of the IHPE, always difficult to order, laboratory, the UMR, in which is based in Montpellier and in Perpignan. It is about epigenetics, and in his abstract, he talks about epigenetics as the rebellious sister. So you can very well imagine that this rebellious sister, it's a bit uncomfortable because indeed challenging this beautiful new synthesis between Mendelian genetics and Darwinism. But from the CMAP, so I'm directing the CMAP LabX in Montpellier, which will still go on until the end of 2024. Several years ago, we really wanted to give a voice to this rebellious sister, and we organized in 2016 a journée scientifique where discussion was engaged, and I think it was really successful in a way that a couple of years later we see this also translated in common projects which have been developed by other CMAP laboratories together with the lab of Christoph. So we had 14 last year. We had a restitution of 14 projets exploratoires, and from these 14 projects, there were actually 3 that had really made use of this epigenetics and clearly integrated in the approach. So we had a nice small session about this subject, I think. So it is taking off the ground. I think this is what Christoph will show us. But then this rebellious system, maybe we also have to remind that this rebellious system now very active in epigenetics and ecology since 10 years, but she probably has a mother or a father which is much older, and just from the end of the 1930s and actually going back to the epigenetics was starting actually in the developmental biology and embryogenesis approaches. So thank you very much, and it's a pleasure to hand over the microphone to Christoph, and please.
Thank you, Roger. And does it work correctly? Hopefully, yeah. Okay, great. So thank you, Roger. Thank you, everybody from the CMAP organizers. Thank you for being here in the room. Thank you. Be over there on Zoom. So, and thank you, Rutger, for putting a real high pressure on me here. I'm really grateful for being here. So it's a big honor to be in this very prestigious seminar, and for me an occasion to reflect on these 10 years of epigenetics and ecology and evolution. And why 10 years? Because almost 10 years ago now we started a research network here in France funded by the CNRS, so the RTP, which continued as a GDR3, and we had really a huge support from not only from the CNRS but from the LabX CMAP. And it's the occasion to engage a discussion. Hopefully I will spend all the 45 minutes, but hopefully we have a nice discussion on these topics. Now, a long time ago, like 10 years or so, when you put epigenetics, ecology, and evolution into the same sentence, that was quite an explosive mixture. And I recall some situation, well, it didn't end up literally like this, discussions between geneticists and epigeneticists, but almost. And I used to say, and maybe I should still say it, I come in peace. Now, I know there are many geneticists in the room, and I'm not saying that epigenetics is a solution to everything, but I think we used these networks of about 150 individual members now to reconciliate genetics and epigenetics in order to better explain what we observe in nature and maybe especially in development. Now, and so by almost Christmas 2013, so the angel of peace came down, sent by the CNRS to make talk together different communities, so geneticists, molecular biologists, embryologists, ecologists, evolutionary science, but also rapidly joined in philosophers, anthropologists, medical doctors to discuss about this link between epigenetics, genetics, and ecology and evolution. So that's a timeline. I realize it's not really visible, so it starts 2013 on the left and now 2023. Don't worry, I will not go through all the details here, but what I put in bold here, that was what I believe is retrospectively or were retrospectively the most important measures. Of course, there's a foundation meeting which happened in Paris, but very rapidly we realized in this network there was this really strong need for training. And this training, for this training, we profited from this École thématique, so summer school possibility of the CNRS, and we organized 3 summer schools, the last one in collaboration, very nice collaboration with the INRAE in Bordeaux. I will come back to this. And then you see there's this cahier prospective. So we participated in the prospectives of the INE and with many colleagues wrote a little leaflet. And we're currently in the process of publishing a book on epigenetics. So with many colleagues and also supported by the LabEx. So of course, other measures like colloques and meetings and special issues and so on. But I really think when I look back over these 10 years, the 4 most important lessons for me is to generate a clear semantics, to have a clear picture of what we are talking about. Well, that's probably true for any field in science, but it was really necessary for epigenetics. Then the importance of training in these concepts, but also training in really solid, down to the earth method that works in any hand. You know, you can give it to anybody and it's going to work. Then third point, make this wet bench technique, meaning molecular biology and bioinformatics together, working together. I mean, ecologists are really excellent in designing experiments and doing sampling strategies and collecting these samples and then bring it together with the molecular biology and bioinformatics systematically. And finally, there is still a strong need for textbooks, especially French textbooks, for students, for undergraduates, for beginners. So anybody come to the lab, you can give this book and he or she can start to work. Now I will walk you through these 4 points and see how we hopefully manage to solve the problems. So generate clear semantics, and that was sort of fire and extinguisher for this explosive mixture to say, what really is epigenetics? And there was a huge work of the members Antoine Nicoglou and Francesca Merlin, who wrote this publication in 2017. And that's a table from their publication, and they came up with the conclusion that there are actually 6 definitions of epigenetics, and it depends on the context. And very often these third definition based on Ricks and Holliday's work in the 70s, so mitotically and meiotically heritable changes in gene function, so not gene expression, that cannot be explained by changes in DNA sequence. So there you have this term of heritability, which is important. But there's also the molecular epigenetics definition, a chromatin modification affecting gene expression, whether it is heritable or not. So all these definitions are absolutely legitimate, but they depend on the context. And when you talk about it, you have to define it. So please, if you use epigenetics in one of your publications, define somewhere what you're talking about. Unfortunately, this publication is behind a paywall, and I took the liberty to put it on the webpage of the network, and I hope I stay under the radar of Elsevier so you can access it. I think that was an important point to define these, and still a couple of years later, I still realize when I talk to people about epigenetics, many of them don't know what it is really. So I send them back to the publication and solves many issues. So based on these definitions, now we put forward with a couple of colleagues the idea that we should update this equation. You know, of course, it comes from Bowman in the 70s. So G cross E gives the phenotype. But when Bowman introduced it, it was still the genotype definition of Johansen from 1909, and which was based on heritability. No molecular biology in 1909. Now, what we propose is to replace genotype by inheritance system. And in this inheritance system, you have different components. There's the genotype, epigenotype, cytoplastic elements, the holobiont, and maybe others. And all these, they interact with each other, and they interact with the environment to bring about in a process the phenotype. So I think that really avoids a potential conflict, and they have an operational definition of genotype, which is now an inheritance system. So that was a couple of years ago, and I was quite pleased to see that finally last year, systems biologist did a bit the same, and it was published in PLOS Biology, and he tried to unify these different things of genetics and epigenetics and what I would call an inheritance system. Now, I think that were two important things, and it evolved very much basically on the semantics, how you use these terms to explain observations. And then there's a second thing in epigenetics to know. Of course, there's a physical basis. There are bearers of epigenetic information that, of course, you know. I think now it's textbook knowledge. You have the interface, the nuclear interface topography, non-coding RNA, histones and other proteins and their modifications, and of course, DNA methylation. Well, but something to remember is that only 3 of them are really linked to the chromosome. So they must stay in the nucleus. That's chromosome linked or chromosomal epigenetic information. But non-coding RNA is extra chromosomal, so it's mobile. It can go outside the nucleus, it can go outside the cell, and it can even go inside another organism. So it's a mobile information that is, for instance, transmitted through the oocyte, but also through the sperm. Okay. Now, second point was a training. As I said, it's really, take-home messages from the last 10 years. So we did these 3 summer schools and quite a lot of training with the CNRS, and we had also the privilege to organize one of these practical classes quite early with a CIMAP. And the summary of what people ask us is finally always, they ask, what exactly is epigenetics? So even if we repeated it several times, we will always explain what is epigenetics in the beginning and always say there are different definitions and it depends on the context. So be clear about what you're talking about. And then the next take-home message is that you need reliable, affordable techniques for non-model organisms. So something that work in practically any context. And on a technical point, finally, people are interested in DNA methylation, and the best method is still something that's called bisulfite sequencing. It converts all the unmethylated cytosines into thymines and all the methylated cytosines into cytosines. and then it converts, therefore, the epigenetic polymorphism into a genetic polymorphism that you can analyze with any method that is available. Next is a TACSEC. So that is an assay for chromatin accessibility, and I will come back to this later. And something that is also systematically requested is to how do I analyze the small or the non-coding RNA in my sample? And many people are interested in transposable elements, naturally doing a link between genetic polymorphism and epigenetic polymorphism. So that's on the methodology part, and bioinformatics should always be included into the package. You cannot separate it one from the other. So those are my experience on these trainings. So training in concepts and techniques. And just to show how this works, so for instance, this was the second summer school. The third one was in winter 2022 with the INRAE, again on a TACSEC, and that time on MobileomeSec. I will say a few words about a TACSEC, but because that attracts really a lot of interest, and it might be interesting to some of you because it's a very straightforward and very available technique. So we thought that in the second summer school on a model which is Daphnia, and the last summer school, everybody could come with her or his most favorite organism and do the TACSEC right away during this school. How does it work? On top you have the chromatin structure, and very oversimplified, you have a closed structure and an open structure. And there is originally bacterial enzyme, which is called TN5. It's a transposase, which is now purified, and adapters, artificial DNA sequences are loaded on this. You incubate your chromatin with it, and the TN5 will integrate in these sites that are accessible to it. The other sites, they are blocked either because they are compacted or maybe because there are some active transcription factors sitting on it. Now, only the regions that are accessible are shredded and amplified by PCR because the adapters are loaded in the same time. Then you sequence it aligned to the genome, and where you have sequences obtained, there is a difference in chromatin structure compared to those that are not. So it's the ATAC stands for Assay for TN5 Accessible Chromatin. So something that proved quite successful to implicate everybody was to write a paper together with practically all the members of the summer school. So as I said, we use Daphnia. So every member had a little Daphnia to pick and to do the TACSEC with it. And the analysis was done by a small group. And actually we repeated an experiment, a very old experiment. So coming from the 1920s, 1930s, you know, Daphnia is very plastic. If you expose it to environmental cues like heavy metals or heat or predators, then it will change the phenotype. And we did an experiment where we had a control water tank and a water tank where a predator was there, but without predation, so just the cues of the predator. And that changed the phenotype, so makes them grow defensive spikes, which we could measure by the length ratio. And on the right-hand side, so it's not very contrasted here on this screen, but it's the correlation between the samples. So if samples have the same epigenotype based on TACSEC data, they are close, so have a zero distance. And if they are very different, they have a longer distance. And what you should see here, but it's difficult, so practically all the stress treatment and all the control treatments, they group together simply based on chromatin structure data in this very homogeneous, genetically homogeneous population. So it's really something straightforward. It meets the requirement of population epigenetics, so it can be done relatively easily. We have a very good feedback so far. And we used heavily also in these training sessions the Epigenome facility we set up with a lot of help from CEMAP, so it's a platform. And just to tell you rapidly how this works, so it's project generation, then we have the different techniques like immunoprecipitation, analysis of DNA methylation by bisulfite sequencing, TechSec available. Much of it is automatized, can be sequenced in an associated sequencing facility or anywhere else where you want. And then there's data analysis. And as I said, it must be in a package. It's our experience. If you don't know how to analyze your data, you end up with nothing. And it's actually sometimes quite difficult to find the right way to produce. Producing the data is today relatively easy. So the analyzers must feed back the wet bench part. And another point that was important and maybe interesting when you set up these facilities, but finally it's quite trivial. Human resource is always a problem, you know, about this. So what we invested is into automatization. So there's a nice robot that does a lot of work, and it allows you to have a very short training period. And so the data generation is, let's say, 80% automatized. Now in France, you have now similar platforms. So they are more focused on human epigenetics and plant epigenetics and ours. And then when you are interested in a particular method, then on the Repertoire of France Genomic, you have more genetics platform that offer also some epigenetic services. That's something good for our community. So this part is taken over by some facilities, and one should not hesitate to contact these facilities for technical support. And to end this bit technical part is a bioinformatics. So what we use is Galaxy as an interface because it's easy to access, it's accessible remotely. And basically we train the colleagues coming on the platform for a couple of days, and then they can do, most of them at least, the analysis remotely. I will come back to some of these challenges on this part a bit later. And also luckily, since last year, there is a Galaxy of the, so the big French bioinformatics network, and it's really powerful. So don't worry about calculation firepower here. Or Galaxy, use Galaxy FR. And finally, so the need for textbooks. So we profited from this possibility, the perspective of the CNRS INE, to produce this little leaflet, which you can find on our webpage, which is actually designed. There's a lot of political, so you can go through it rapidly, but there's a lot of things I give, for instance, my students, and it's really written for undergraduate students and beginners. So it's a nice resource, and it's free, and it's in French, but it was, of course, not enough. And so we started almost 2 years ago now to write with a number of colleagues for the ESD edition, this Explique-moi l'épigénétique, explain me epigenetics, and we treat, so of course, the historical and epistemological concepts, molecular epigenetics, epigenetics and transposable elements, but also epigenetics and plasticity, adaptation, some very precise examples in response to climate change, epigenetics and evolution, and also the concepts that occur in the society now about epigenetics, also sometimes misconceptions and false hopes, but also things you can exploit now in terms of epigenetics. And so I'd like to thank the SEM again, who helped to organize this first meeting. And we started to write this together, and now it's in the hand of ESTE, and we hope it will be published very, very soon. Okay, that was large overview, but just to talk a little bit also about the most recent results and the outlook. So my first request in the outlook, and I know I'm addressing myself to the right community, geneticists, please define and redefine genetics. I think it's a real work to do, and maybe it's a point of discussion later. Another challenge is data aggregation. We have now many data on proteomics, RNA-seq, epigenetics, genetics. We have to bring all this together, and there is still a lot of work to do, and maybe there are also people in the room or over there on Zoom who would like to contribute. Epigenetic engineering, you mean changing specifically a locus in its chromatin structure or DNA methylation and see what phenotype it produces. And that is something I will talk about. And I personally think that the social perception, the commercial use of epigenetics will be something we should really be concerned about, not worried, but we should, I think, prepared to it. And if you go a little bit further to the south, for instance, this is a picture from Barcelona on a pharmacy. You can go in Barcelona to a pharmacy and get your epigenetics profile. So now, what does it tell you? It's not allowed in France, but I mean, you take your car, you have to 250 kilometers, and you have your epigenetic profile. You can go do it online. You know, you can send it to the US, and you have an epigenetic profiling of yourself. And of course, single cell techniques, that's on the technical part. I wanted to talk about epigenetic engineering because I think this is something that can really answer still the question, so how does epigenetic work finally? You know, how does it translate, epigenetic change translate into a phenotypic change? Now, a couple of years, we started a collaboration with Margaret Godel. She developed a DEDCAS-based targeted DNA methylation method. In vertebrates, there was this strong hypothesis that if you have hypomethylation in a promoter, then you can have transcription, given there's all the other factors available. And if it's hypermethylated, so overmethylated in the promoter, then the gene is silent. So it's like a functional deletion. And she designs a system in which she used DEDCAS, so it doesn't cut, but it is still guided by guide RNA and can be associated with an methyltransferase catalytic activity to specifically methylated a promoter region, and that's the result is shown here on the left. You have the black bar and the white, which is not visible here, but they're almost the same size. These are the controls. So this is the expression of this specific gene. And you have the red bar that is after exposure to the system which methylated the promoter region. So that was done in cell culture. And we wanted to do it in a real living organism. And again, I apologize for the contrast here. That was one of our PhD students, Nelia Loviana, who did all the work. Yeah, so please, well, focus on the blue and the yellow. It's a double system where you have on the left-hand side DCAS9, so DEDCAS, it doesn't cut, it's still guided, associated with the blue fluorescence protein, so you can trace it. On the right side, you have DNA methyltransferase catalytic activity of humans of 3A, GFP to trace it. And both can interact through these fragments, antibody fragments that specifically detect these brown little bits, which are called SANTAC. And when it comes together, it will recognize a locus on the DNA, amplify the amount of DNMT3A you have associated, and will methylate the associated locus. So we did this in a small snail, Biomphalaria glabrata, which is a vector of an important disease. That's why we're interested in. By micro-injecting these plasmids into the embryonic stages, what you see here from the left to the right and then down panel are the first cleavage stages of the embryos. So on the left-hand side is a fertilized eggs. And actually the embryo is a little brown dot you see. And the particularity of the snail is that the eggs are laid in patches, and what you see here is this egg patch. Now that worked, and we could transfect, again, I will not go into the details, at 72-hour stages with GFP, with blue fluorescence protein. And so we had embryos that developed having both signals, meaning that we had expression of both plasmids, and to test then if we had also the expression, at least transcription of the enzymatic activity of DNA methylation and Cas9, we did quantitative PCR, RT-PCR. On the left-hand side, the controls, 0, of course, and on the right-hand side, each time the qPCR, qRT-PCR. There are different snails because they're so small, we cannot do all the same experiment on the same snail. And what we could show that, so we did the transfection at day 4, or at day, sorry, at day 1. Then the expression of the protein started, so by fluorescence in day 4, which is quite logic. You have to produce the protein, start transcription, express the protein. At that time, we injected the guide RNA because we could detect the positive snails. Then expression of the protein continued for 4 days. And then just before hatching, luckily, no more fluorescence, so supposedly no more activity of this methylation, which is something that is desired. You don't want to have this methylation activity all the time. It's an approach that is called hit and run. You inject and then you lose the activity. And so we had 4 snails on the right-hand side. The red bars show the mean methylation at this target locus. On the left-hand side in blue, the controls. And what you see, there are 4 snails, whereas quite strong change in DNA methylation. Summary, and I'm almost finished. So the system works. Also in our snail, targeted methylation is possible. Efficiency is quite low, so it's 2%. 4 positives out of 250, but it's reasonable, and we have ideas how to improve it. And it was actually the first time that was done in a mollusk, and only the second time in an invertebrate. And given that so many people are interested in invertebrates and the importance of invertebrates, it shows us also a strong bias, in these techniques towards a too strong bias, in my opinion, towards model organism, and which comes back to training and concept and techniques. Yeah, thank you very much for listening. I'm happy to take questions. I'm very grateful to the CEMEP, Séverine Chamberon, who is the second director of this GDR, which probably will finish this year. We will see if the young people are interested. It might continue. Thank you.
Okay, thank you very much, Christoph. Any questions in the room? Yeah.
Thank you very much for the talk and for setting this group. I guess it was a big, it's been a big adventure, I guess. So I'm not allowed into, I don't have knowledge of epigenetics, and it's been, of course, as many a word I hear and I try to grasp in my experiments or in my models, how to implement it.
Yeah.
And the first part you talked about defining it. So if there are 6 different, for example, and maybe there are sub-branches in those, I don't know. Yeah. Is there now more of a consensus on not on the mechanism, things like methylation, etc., but on more things that, as an evolutionary biologist, I would be interested in is more as a transmitted acquired characters, because they exist. Of course, language is transmitted acquired characters. So I have trouble distinguishing plasticity and epigenetics, gene regulation and epigenetics. What is different between a hormone that modifies my whatever in my body if I inject it, but I'm genetically coded to respond to that hormone that way? That's I guess it's a question you often have.
Yeah, perfect. So these are precisely the questions to ask. And I think that the definitions do not answer the question, but they set the scene, you know. And for instance, I would say there are 3 main écoles de pensée. It's the anything that changes chromatin structure is epigenetic. And it has an effect on a gene or whatever, who cares? So that's very molecular. Then second, it must be heritable. So it's heritable changes which might or might not influence gene function, but without changes in DNA sequence. And there's this term heritability, and this heritability you can measure, of course, like by classical means. And you can also measure the heritability of DNA methylation, which is now relatively easy to do. So, and if the second will have evolutionary consequences in the sense, if you produce a heritable change in a phenotype, then selection can act on this phenotype. Depending on the, sort of course, you know, and so depending on the selection pressure and the heritability, you will have evolutionary consequences. And then you have let's say the very molecular school of thinking where you say, well, we are interested in the mechanism. These changes can be heritable or not, who cares? I want to know how whatever histone modification influences on gene expression. I wouldn't call it regulation, except for maybe Plasmodium, where you have almost no transcription factors, but I would call it control rather of gene expression. But you are right, there's a big mixture, and even, yeah, so there are 3 types of publications. The good publications give definition. The bad publications, they don't give definitions. And the very bad, they mix everything, you know, and but you find all of them in the literature and in very good journals. So I think when you start, you said, I don't know anything about epigenetics, which is probably wrong, because you know already a lot about epigenetics, but it was not necessarily called epigenetic also. So when you know the work of Barbara McClintock, for instance, there is epigenetics inside, but she didn't use the word. There's also a bit of semantic confusion. Yeah, so I really would like to read this publication. Okay, there are these different definitions, and depending on your context, they are all legitimate, I would say.
Okay, merci. You want to add something, or is there another question? Also, this question from Christoph. Somebody would like to redefine genetics.
Oh yes,
was a bit provocative, maybe.
Yeah, but not enough. Maybe
the challenge in the chat. Yes.
No challenge, but more like a naive question. So something or someone must control the putting of the methylation. Yeah, I suppose genes. So are we then not back to genetics?
Yeah. So if I would, I will answer the question, but I could ask you, what is a gene? Yeah. So it brings me back to the systems idea. So there is no genome without epigenome. Doesn't exist. It exists in your test tube, but nowhere else. And there's no epigenome without genome. So in nature, in your cell, they are both together. So they interact. Now, who controls the setting of epigenetic marks? Of course, you need genes that code for proteins. You need a catalytic activity, for instance. You need all the machinery that is necessary to establish the epigenetic marks. But it is not enough. And that you see, for instance, for cloning animals. No. What you need is also epigenetic information. And we know now, well, the big question was, and it is not entirely solved, is how is this information transmitted in the first place? And I think most, well, if you, the information about mitotic heritability is based on the chromatin structure itself. This is quite clear. So it's DNA methylation that guides DNA methylation in the next mitotic generation, and its chromatin structure is a bit more complicated, involving other proteins like heterochromatin 1, etc., which determines the chromatin structure of the next mitotic generation. The question was about meiotic generation. You will know that every gametogenesis, there's a reset of the epigenetic information, different waves of demethylation, remethylation, for instance, which is very well analyzed. Histones are replaced by proteamines, etc. We know now that's not completely erased first. So some of the DNA methylation stays, some of the histone modification stays, and there's a lot of non-coding RNA that is involved, which is used to establish the positions of the chromatin structures. And at the same time, the epigenome will influence on mutation rate, for instance. And of course, you need the genome to establish an epigenome. No? So it's not it's a egg and hen problem, and both act together. So I would say what is more important than the others is a wrong question, actually.
Okay, thank you. Maybe you would like to add something, or
so how would you define a gene? It's not a trick. It's really I mean, I have heard so many definitions.
Okay. You had a question? No?
So I want to clarify the question. It's not risky. Yeah, so when you ask define a gene, so define genetics, and maybe before that define a gene, meaning because there are definitions of genes like that were being taught in school. So like I guess gene from memory would be a unit that encodes for a protein, maybe a sequence that encodes for a protein, but there will be of course maturation, there will be etc. But one gene is something, and it has pleiotropic effects, and some genes can be regulators of other genes, but there is kind of a unit in molecular evolution. They take an initiator codon, a stop codon. They have kind of so what is the problematic that requires what is the particular thing that, I guess, in the light of epigenetics?
I think it's about, well, I think you had two things in your definition. You had unit of heritability and you had protein coding. So can you already say, well, what about long non-coding RNA? Is it coded by a gene or not gene? No.
It's not acting. They don't call it gene, but it's all the question about the dump DNA that has factors affecting. What is the difference between a lactose operon in a bacteria and a piece of non-coding DNA that can, comment dire, se replier and affect the expression of others? But it still is something that I inherit in the classic Mendelian way, kind of.
Okay.
And that affects the expression of the other genes. I don't know.
I'm feeling uncomfortable, but I mean, that's a really open discussion. I think I'm feeling uncomfortable with the molecular definition plus heritable. But if you think, let's take the molecular definition. Okay, well, it's a piece of DNA that codes for a protein, basically. Well, okay, let's think it through. So we only take proteins, but we now know now in our cells, there are more long non-coding, so not messenger RNAs that mRNAs. Okay, so let's include long non-coding RNAs into the gene. Well, then we have all these small RNA and we have also viruses and so on. Okay, is this a gene or not? So let's include this, which finally brought up that all of the genome is coding for something. So all is a gene, which is a very, yeah, I would say, is it really useful as a definition of a gene? And then heritability. So you're talking heritability. What heritability? Is it transmission through the germline from one generation to the other, I suppose, with a heritability of 1? But you have very stable epimutations that are inherited the same way that was demonstrated and which produce the same phenotype. And in this sense, a gene
heritability in the quantitative genetics, like a part of variance, NANOVA kind of definition, part of variance that is explained by differences in.
But variance, you're talking about DNA changes, mutations, DNA mutations. So if you restrict, then you restrict it to everything that is linked to DNA only. You know, basically you say genetics is the science of DNA. No. If quantitative genetics very often uses the genetic polymorphisms without necessarily knowing if there is any effect as a measure of genetic polymorphism, which is absolutely legitimate, yeah, you know. But then we restrict somehow genetics, the definition of genetics, to genetic polymorphism. And we have already, in this sense, 3 definitions of genetics, which are absolutely legitimate, I would say, which is okay. I mean, but if you talk to an epigeneticist, I think you have to be clear about what you want to know. You want to have a functional impact, which is heritable. Then if you have only heritability patterns, you don't know if it's genetic or epigenetic. No.
Is there a distinction because the way it's transmitted is different? I mean, if I have a sexual genome, I have the infinitesimal model, it's diluted, blah, blah, blah. There are simple rules that may or not be valid, but they have been tested in the centuries in quantitative genetics. And so I would have expected that this would be like the new model, and the deviations from that are ways to
it's a way to
another way of being transmitted and diluted and mutating and all that.
It's a way to do it. So you have genome-wide association studies, epigenome-wide association studies, and then now sophisticated models I don't understand at all that manage to disentangle both and that can quantify what is DNA genetic, well, genetic polymorphism, which epigenetic polymorphism is determined by a genetic polymorphism that of course that exists, and what is purely epigenetic, but that's disentangled mathematically by statistical approaches based on differences in heritability.
Thank you. I saw that Patrice had a question and maybe some others afterwards.
Yeah, I think in part we are having in epigenetics the same kind of trajectory that genetics has had before. So when we think about the birth of genetics, so the idea was just heritable changes. And so this tradition gave rise to what we call today quantitative genetics, in which we model the heredity of traits. And we actually don't, I mean, the quantitative genetic approach doesn't impose that everything that's transmitted and modeled by the quantitative genetic model is on the DNA sequence. you could incorporate anything. So it's not because we did some fancy models like the infinitesimal one that can link together Mendelian sequence-based inheritance to quantitative genetics. It's not because of this that quantitative genetics doesn't take everything on board, including what we would now call inherited epigenetic changes. So that's the first tradition. And then in the 60s, we have had molecular biology, and so everything became DNA sequence. And so there's been this kind of reduction in which we take the Mendelian model and down to the molecular level and reduce it to change in sequence. So this has become the genetics that epigeneticists use to delimitate. It's based on the DNA sequence in terms of nucleotides, ATGC, and everything else, which is not in the domain of molecular genetics, we will call it epigenetics, which is this, I think, one of the definitions you gave, the molecular one. And I think we are down to this. In molecular terms, we have a simple way to tease apart one domain from the other. One is concerned with the DNA sequence ATGC, not including methylations. And the other one is concerned with everything else that carries information from one cell to the daughter cell, and possibly from one generation to the other generation, if it goes through the germline. But I think what people have settled on is that it's only by the nature of the molecular mechanism that we distinguish both.
I agree with you. But if you said it like this, I mean, it's very clear. When you start about, many people include, sometimes I hear if it can be influenced by the environment, it's epigenetic, which is true and wrong. I mean, take the example of CRISPR-Cas, you can very nicely influence by the environment on the genome as well. And this is only one of the examples. So, yeah, so I agree with you, but you have defined, the frontiers or the two elements, genetic, epigenetic, on a common basis. This is also important. I think a mistake that I've seen from time to time that sometimes, one element is defined by its heritability. So Mendelian heritability is genetic, and then we talk about histone modification. So it's epigenetic. So we are on two completely different scales and bases of definition. So that creates problems in the end. Just Jacob Monod in their publications, I don't remember exactly how they said it, but sort of, it's clear now what is a gene. So let's continue to, they didn't even bother about defining what a gene is. So they continued straight away to the operon. And I find it interesting. And at that time, it was clear. I think it was clear to them that it is based on DNA, but they're dead, so I cannot ask them anymore.
Okay, thank you. Any other questions? Maybe in the chat? No? Here in the room? Still, anybody would like to redefine genetics after this discussion in another way, maybe? No? Otherwise, we thank you all very much, and particularly thank Christoph for his nice presentation and for coming to Montpellier for this occasion.
Thank you.